The field of spatial genomics has emerged as one of the most transformative frontiers in biological and medical research, offering a window into the intricate tapestry of gene expression within the native context of tissues and organs. Traditional genomics and transcriptomics have provided invaluable insights by cataloging the genetic and transcriptional content of cells, yet they often do so at the cost of spatial information. By isolating cells from their environments, these approaches inherently lose the contextual cues, cellular neighborhoods, tissue architecture, and microenvironmental influences that are essential for understanding the true complexity of biological systems. Spatial genomics bridges this critical gap, enabling researchers to map where genes are being expressed within intact tissues and thus unraveling the spatial relationships that underlie development, health, and disease. At its core, spatial genomics encompasses a suite of technologies and analytical strategies that allow scientists to visualize and quantify gene expression, DNA, or other molecular features with precise spatial resolution. The field draws on advances in molecular biology, imaging, microfluidics, and computational analysis, blending them into powerful platforms capable of interrogating tissues at single-cell or even subcellular resolution. Early efforts in spatial genomics relied heavily on in situ hybridization techniques, which use labeled probes to detect specific RNA or DNA sequences in fixed tissue sections. While these methods laid the groundwork, they were limited by low throughput and the number of targets that could be simultaneously analyzed. The advent of high-throughput sequencing and multiplexed imaging has revolutionized spatial genomics. Methods such as spatial transcriptomics, multiplexed error-robust fluorescence in situ hybridization, MERFISH, sequential fluorescence in situ hybridization, SecuFISH, SlideSeq, and 10x Genomics' Visium platform have dramatically expanded the scale and depth at which spatial gene expression can be profiled. For example, spatial transcriptomics enables the capture of spatially resolved RNA profiles across thousands of spots on a tissue section, each representing a small group of cells. Meanwhile, highly multiplexed imaging methods can visualize hundreds to thousands of RNA molecules or protein markers within single cells, preserving their spatial context with exquisite detail. These technological innovations have unlocked unprecedented opportunities to explore fundamental questions in biology. In developmental biology, spatial genomics allows researchers to trace how gene expression patterns orchestrate the emergence of complex tissues and organs from undifferentiated progenitors. By mapping the spatial dynamics of transcripts and regulatory elements during embryogenesis, scientists can reconstruct lineage relationships and identify the signaling centers that drive pattern formation. Similarly, in neuroscience, spatial genomics has illuminated the cellular diversity of the brain, revealing how distinct neuronal and glial populations are organized within functional circuits and how these architectures are perturbed in neurodevelopmental and neurodegenerative disorders. Cancer research has been profoundly impacted by spatial genomics, as tumors are inherently heterogeneous ecosystems composed of malignant cells, stromal components, immune infiltrates, and vasculature. Bulk sequencing approaches often obscure this heterogeneity, averaging out signals from diverse cell types and microenvironments. Spatial genomics overcomes this limitation, enabling the dissection of tumor architecture at single-cell resolution. Researchers can now map the spatial distribution of cancer cells, immune cells, and stromal elements, uncovering how their interactions shape tumor progression, therapeutic response, and resistance. For example, spatial transcriptomics has revealed immune hot and cold regions within tumors, correlating these landscapes with patient outcomes and informing strategies for immunotherapy. The power of spatial genomics extends beyond basic research, holding immense promise for translational medicine and diagnostics. In pathology, spatially resolved molecular profiling augments traditional histology, providing a molecular atlas that can distinguish subtle disease subtypes, predict prognosis, or guide targeted therapies. For instance, spatial genomics can identify early molecular changes in pre-malignant lesions, map the spread of infectious agents within tissues, or monitor the response of individual cells to treatment. As these technologies become more accessible and standardized, it is conceivable that spatial genomics will become an integral component of clinical workflows, driving precision medicine to new heights. Despite its transformative potential, the field of spatial genomics faces significant technical and analytical challenges. One major hurdle lies in balancing resolution, throughput, and molecular coverage. High-resolution imaging methods can achieve subcellular detail, but are often limited in the number of genes or proteins they can measure simultaneously. Conversely, sequencing-based methods offer broader coverage, but may sacrifice spatial resolution or require complex barcoding schemes. Integrating these complementary approaches, imaging and sequencing, is an active area of research, with hybrid platforms aiming to deliver both comprehensive molecular profiles and fine-grained spatial maps. Data analysis and interpretation represent another formidable challenge. Spatial genomics experiments generate vast, high-dimensional datasets that combine molecular measurements with spatial coordinates, often across large tissue sections or entire organs. Extracting meaningful biological insights from such data requires sophisticated computational tools for image processing, spatial statistics, data integration, and visualization. Machine learning and artificial intelligence are increasingly employed to identify spatial patterns, classify cell types, infer cell-cell interactions, and reconstruct tissue architectures. The development of robust, user-friendly software pipelines is essential to democratize access to spatial genomics and enable widespread adoption across diverse research settings. Standardization and reproducibility are also critical considerations as spatial genomics matures. Variability in sample preparation, imaging, sequencing, and data analysis can introduce batch effects and confound biological interpretations. Efforts are underway to establish best practices, reference materials, and benchmarking datasets to ensure consistency and reliability across laboratories. Collaborative initiatives and consortia, such as the Human Cell Atlas and the NIH-funded HubMap project, are driving the creation of comprehensive spatial maps of human tissues, fostering data sharing and methodological innovation. Ethical, legal, and social implications accompany the rapid growth of spatial genomics, particularly as it moves closer to clinical application. The ability to generate detailed molecular maps of human tissues raises questions about privacy, consent, and data security. As with other omics technologies, it is essential to engage stakeholders, including patients, clinicians, ethicists, and policymakers, in discussions about the responsible use of spatial genomics data, ensuring that benefits are equitably distributed and risks are appropriately managed. Looking ahead, the future of spatial genomics is bright, with several exciting directions on the horizon. Advances in probe design, imaging hardware, and sequencing chemistry are expected to further increase the sensitivity, specificity, and multiplexing capacity of spatial genomics platforms. Integration with other omics modalities, such as proteomics, metabolomics, and epigenomics, will enable multi-layered spatial analyses that capture the full complexity of cellular states and interactions. Three-dimensional, and even four-dimensional, spatiotemporal mapping of tissues will provide dynamic views of biological processes as they unfold over time. Single-cell spatial genomics is poised to play a pivotal role in the emerging field of systems biology, where the goal is to model and predict the behavior of complex biological systems based on their constituent parts and interactions. By providing quantitative, spatially resolved data on gene expression and cell organization, spatial genomics offers a foundation for constructing accurate models of tissue function, development, and disease. Such models have the potential to revolutionize drug discovery, regenerative medicine, and synthetic biology, enabling the rational design of interventions that target specific cells or microenvironments within tissues. In the realm of developmental biology, spatial genomics is enabling a renaissance in our understanding of how multicellular organisms are built from single cells. By combining lineage tracing, spatial transcriptomics, and live imaging, researchers can reconstruct the trajectories of individual cells as they differentiate, migrate, and assemble into functional tissues. These insights are shedding light on the fundamental principles of morphogenesis, pattern formation, and organogenesis, with implications for congenital disease, tissue engineering, and regenerative therapies. Neuroscience stands to benefit immensely from spatial genomics, given the extraordinary cellular diversity and complexity of the nervous system. Mapping the molecular identities and spatial relationships of neurons, glia, and other cell types across brain regions is crucial for understanding how neural circuits are wired and how they malfunction in disease. Spatial genomics has already revealed novel cell types, uncovered region-specific transcriptional programs, and identified spatial gradients of gene expression that underpin brain function. As methods continue to improve, it is likely that spatial genomics will illuminate the molecular basis of cognition, behavior, and neuropsychiatric disorders. In cancer biology, spatial genomics is transforming our understanding of tumor ecosystems. Tumors are not simply masses of proliferating cells. They are dynamic communities shaped by interactions among cancer cells, immune cells, stromal elements, and the extracellular matrix. Spatial genomics provides a lens through which to observe these interactions in situ, identifying niches that support tumor growth, regions of immune exclusion or activation, and pathways of metastasis. Such information is invaluable for designing more effective, personalized therapies that target not only cancer cells, but also their supportive microenvironments. The integration of spatial genomics with digital pathology and artificial intelligence is opening new avenues for automated quantitative tissue analysis. By combining molecular maps with high-resolution histological images, pathologists can gain deeper insights into disease mechanisms, stratify patients with greater accuracy, and predict therapeutic responses. AI-driven algorithms can assist in identifying spatial biomarkers, segmenting tissue structures, and correlating molecular features with clinical outcomes. This convergence of spatial genomics, imaging, and computational analysis heralds a new era of precision diagnostics and prognostics. Spatial genomics is also making inroads into immunology, where the spatial organization of immune cells within tissues is critical for mounting effective responses to infection, injury, or malignancy. By mapping the locations and phenotypes of immune cells in lymphoid organs, tumors, or sites of inflammation, researchers can decipher the rules governing immune surveillance, tolerance, and memory. These insights have direct implications for vaccine development, immunotherapy, and the treatment of autoimmune diseases. Beyond human health, spatial genomics is being applied to a wide range of organisms and ecological contexts. In plant biology, spatial transcriptomics is elucidating how gene expression patterns shape tissue differentiation, organ development, and responses to environmental stress. In microbiology, spatial genomics is uncovering the organization and function of microbial communities within host tissues or environmental niches. These studies are expanding our understanding of the fundamental principles that govern life across scales and species. Education and training in spatial genomics are becoming increasingly important as the field grows. Researchers must acquire interdisciplinary skills, spanning molecular biology, imaging, bioinformatics, and computational modeling. Academic institutions, research consortia, and industry partners are developing courses, workshops, and online resources to equip the next generation of scientists with the tools and knowledge needed to harness the power of spatial genomics. Collaboration is a hallmark of the spatial genomics community, as the complexity of the field demands expertise from multiple disciplines. Biologists, engineers, chemists, physicists, computer scientists, and clinicians are working together to develop new methods, interpret data, and translate findings into practical applications. Large-scale initiatives, such as the Human Cell Atlas, are bringing together thousands of scientists worldwide to create comprehensive, spatially resolved maps of all human cells. These collaborative efforts are generating open-access datasets, protocols, and analytical tools that benefit the entire scientific community. As spatial genomics continues to evolve, it is likely to intersect with other cutting-edge fields, such as synthetic biology, organoid technology, and gene editing. Ultimately, the promise of spatial genomics lies in its ability to reveal the hidden architecture of life. By mapping the molecular landscapes of tissues in situ, spatial genomics allows us to see not only what cells are present, but where they are, how they interact, and how their collective behaviors give rise to health and disease. This holistic perspective is essential for tackling some of the most pressing challenges in biology and medicine, from understanding development and aging to combating cancer, neurodegeneration, and infectious diseases. The journey of spatial genomics is just beginning, and the coming years are likely to witness even more dramatic advances in technology, analysis, and application. As researchers continue to push the boundaries of what is possible, spatial genomics will undoubtedly deepen our understanding of the living world and pave the way for new therapies, diagnostics, and biotechnologies. The field exemplifies the power of interdisciplinary science, driven by curiosity, innovation, and a shared quest. Decipher